Hi, everyone. My name is Arianne. I'm faculty at Arizona State University and the director of the Global Locust Initiative. I wanted to start out by thanking the organizers for putting on this full-day webinar and all the work that goes into that. Unfortunately, due to the time change, for those of us working in the Western US, it's the middle of the night, so we pre-recorded this talk, but we look forward to joining you for the breakout sessions at the end of the day's webinar and listening to everyone else's talks as soon as they become available. I've asked a few team members and partners to contribute short videos, so you'll hear their voices throughout the talk. I also wanted to send a shout out to everyone that's working to address the ongoing locust outbreaks, particularly those of you who are on the ground. It is a lot of hard work, but it's really critical, so thank you. As I'm sure you've already talked about today during the webinar, the short-term impacts on agriculture can be astounding. But something that we talk less about as a global community are the long-term impacts on livelihood. One of the reasons is that there's actually not much research done on this topic, but from a few studies we know that, for example, in West Africa, children born during plague years in villages that are impacted by locusts are much less likely to ever start school. And the point I want to drive home is that locusts don't act in isolation. They are part of a very complex social, ecological, technological system. So here's one diagram that attempts to capture part of that. And here you can see that, for example, there are effects of land use on the probability of locust outbreak. Locusts don't stay locally. They migrate out to distant regions, which is represented by outside of the circle. And indeed, we have to work together across boundaries and sectors, across disciplines, in order to find solutions to the global challenge of locust plagues. To that aim, we created the Global Locust Initiative.
So for those of you who might not know, the Global Locust Initiative is an Arizona State University initiative whose mission is to promote interdisciplinary locust research and management to improve the well-being of farming communities and global food system sustainability. If you haven't visited our website yet, or it's been a while, please visit us at locust.asu.edu to connect with us, to learn more, or to join the initiative. The GLI has two arms, GLI Network and GLI Labs, and I'll talk briefly about both of those. As we're all aware, locusts are a continental problem that requires coordinated response across boundaries and sectors. And the purpose of this first arm of the GLI Network, is to connect universities, government and non-government organizations, and agricultural communities to forge new partnerships that seek to better integrate locust research and management. Our current network contains over 375 members spanning at least 55 countries with expertise in integrative pest management, monitoring and forecasting, grasshopper and locust biology, landscape ecology, food security, social sciences, and many other areas. To borrow a phrase from Chris Adriansen from the Australian Plague Locust Commission, is the courage and confidence it provides when we as members are strengthened and supported by a global network of aligned interest that is larger than our respective institutions or individual nations. And I've seen many great examples of that with the GLI network recently in tandem with these ongoing crises that we're experiencing globally. In the near future, we're working on setting up a platform using a software similar to Slack, for those of you who know what that is, to streamline coordination and communication among members. So look for that development in the near future. The second arm of the GLI is GLI Labs, which hosts a number of research projects spanning natural and social sciences with the aim of advancing our fundamental understanding of the systems in which locusts and grasshoppers exist, as well as supporting the development of local solutions to the global challenge of locust plagues. So GLI Lab supports a number of projects on several continents, which we'll hear more about shortly from GLI members who are leading these respective projects. But I'd like to just briefly mention two exciting new 5-year projects that GLI Labs will be embarking on, both funded by the National Science Foundation of the United States. One of these projects will broadly investigate the ecological and evolutionary trade-offs that locusts face between nutrition, migration, and immunity. And a second project will produce a research institute across several universities, including Baylor College of Medicine, Texas A&M University, and others. And this institute's efforts will be aimed at deepening our understanding of locust phenotypic plasticity from molecules to landscapes. And it will provide ultimately extensive and publicly available molecular resources, such as high-quality annotated genomes and transcriptomes for several locust and grasshopper species, including the desert locust.
Most of our GLI Labs projects focus on understanding how locusts fit into this broader system. And there's an increasing understanding that people are an important part of the system, and we're impacting the system not only through policy and management, but also through our impact on the landscape, so things like changing land use and climate. These all feed back into the system and affect the probability of a locust outbreak locally, regionally, and internationally. Here's an example using the Senegalese grasshopper, Adalia senegalensis. So this species doesn't form swarms anywhere near the magnitude of the desert locust, but it does have some locust-like characteristics in that it aggregates, it can still travel long distances, it crosses country boundaries, and it also has pretty impressive impacts on livelihood. We've been studying this species for a number of years now, and our team and others have shown that there's a strong relationship between land use type and where this species is most abundant. And in particular, it tends to hang out in areas that have plants with low nitrogen content, such as fallow or grazing fields. And Marion's going to talk to you about why.
A little bit about nutritional ecology. So I work with herbivorous insects, and one of the particularity of them is obviously they feed on plants. And one thing that's interesting about plants is that they usually contain a lot less protein than meat does, right? So like usually a challenge for herbivores is securing enough protein. That is why typically there is a strong relationship between nitrogen content of plants and insects' performance. However, we started noticing a couple of years, for several years now, that in the lab, locusts, when you give them the choice between two dishes, they tend to select for dishes that have more sugar than protein, and that is very strange for a herbivore. This is some field work we did in 2017. So we went to a millet field. So millet in Senegal is a subsistence crop. And so we fertilized some plots with 2 levels of fertilizer, and some we left without fertilizer. So those are our control treatments. So basically what we obtain are in the control plant that are like more sugary, and in the protein treatment, in the fertilizer treatment, plant that have more protein. And in the medium, we have an intermediate level. Then we built some cage, as you can see on the picture, and we put locusts in them. We found that unlike what we know for herbivores insects, remember, like the plants are normally pretty poor in protein, they did not do better when we added fertilizer. As a matter of fact, when it comes to female, they actually did worse, right? You can see that like female in the control treatments, the one that received no fertilizers, so the plants that were more sugary, the insects survived better. So that allowed us to collect the eggs that were present in each cage. We found that when there is a high level of protein in the plants, the egg mass was actually lower. One of the reasons could be that if you ever run a marathon, I personally don't run, that's definitely something that's against my religion. But if I were to, I probably would carb load, right? And we think that's what's happening with locusts. So locusts are a very specific type of grasshopper that undergoes migration. You know, they swarm. And migration in insects is actually very energetically costly. They're not very efficient flyers, so they need a lot of reserves. And for grasshopper and locusts, we know that flight energetic are fueled by lipid. And how do you get lipid? How do you get fat? Well, by eating a lot of carbohydrates. So we think that's a mechanism that exists here.
Because this pest is highly migratory, it requires a coordinated response across communities and in some cases across countries. So we partnered with the U.S. Agency for International Development to put together a area-wide integrated pest management approach using soil amendments. Working with the Senegalese Plant Protection Directorate, we developed a community-based early warning system and trained a women's group across 5 different villages, worked with them to put in light traps so that they could track the nightly migrations of this pest, as well as do surveys for them in their fields. We partnered with McGill University, Red Cross, and others to conduct stakeholder interviews to understand how this project impacts livelihoods. And here to share some of the exciting results from the project thus far is our partner Mamour Touré.
Bonjour, moi c'est Dr Mamour Touré de l'Université Gaston Berger de Saint-Louis au Sénégal. Le projet porte sur la surveillance et la gestion des essais décriés par l'amendement du sol. L'objectif principal est d'augmenter les capacités techniques et scientifiques des populations villageoises en termes de surveillance des acridiens par le biais de l'amendement du sol. Nous avons travaillé avec 100 paysans dans 2 villages situés dans les régions de Kaffrine et de Fatick au Sénégal. Chaque paysan possède un champ fertilisé et un champ témoin. Nous avons effectué des prospections en début, en milieu et en fin de saison des pluies pour voir le nombre de criquets et leur impact sur le milieu. Les résultats obtenus ont montré que la fertilisation diminue le nombre de criquets sur le terrain et par conséquent les dommages causés par ces criquets sur le mil. La fertilisation augmente aussi la production de mil car les récoltes sont beaucoup plus importantes avec les champs fertilisés que dans les champs non fertilisés. Nous pouvons retenir donc que la fertilisation a un double impact, diminue considérablement le nombre de criquets sur le mil en créant un environnement défavorable à leur nutrition, mais aussi l'augmentation de la production. Je vous remercie.
As many of you are aware, in addition to the desert locust crisis, there's been an ongoing South American locust outbreak, and many countries over the past few years have seen locust outbreaks on a scale that haven't been seen for more than 60 years. GLI has several partners and projects in the region, and one of the projects is asking similar questions to the research that we just showed you. So this species is pretty closely related to the desert locust, it's Schistosarca cantillata, and we wanted to know if the same nutritional limitations, limitation in carbohydrates specifically, could be applied to this locust as well. So we went out into the field, again working with many excellent partners, and in front of a marching band we put out these different diets that had different levels of protein to carbohydrate content, and what we found over many bands over multiple countries is that most of the locusts selected chose to eat from the diets that were very sugary, that had a high carbohydrate content, and largely ignored dishes that were high in protein. So this suggests that they were carbohydrate limited in the field. And then if we compare what their self-selected nutritional content was relative to the plants that we collected from the various areas where these outbreaks were happening, also corroborates this and suggests that in general these locusts are carbohydrate limited, and that areas that have high carbohydrate, low protein plants are going to fuel long distance migrations. And here is Hector Medina, who leads the locust program for Argentina Senasa, to give another update.
Hola, mi nombre es Héctor Medina. Soy el coordinador del Programa Nacional de Langostas y Saltamontes del SENASA Argentina. También soy el coordinador del Plan Regional de Manejo de la Langosta Sudamericana entre los países de Argentina, Bolivia y Paraguay. A su vez, Soy el coordinador del Grupo Langosta del Grupo Interamericano en Coordinación de Sanidad Vegetal, GIXB, y soy miembro del GLI. Luego de 60 años, con poca actividad de langosta en el continente, la langosta sudamericana tuvo un dramático resurgimiento que comenzó en el año 2015 y persiste hasta el día de hoy. Junto al GLI Team, junto al equipo del GLI, hemos colaborado en experimentos para entender el comportamiento alimentario y la fisiología nutricional de las langostas, tanto en experiencias de campo como en laboratorio en distintos países de nuestra región. Con el GLI, el Instituto Interamericano de Cooperación para la Agricultura, el ICA, y el SENASA Argentina, organizamos un workshop donde intentamos entender la estructura en el manejo de la gobernanza de langostas. Los resultados de este workshop serán comparados con talleres similares que se harán en otras partes del mundo, más precisamente en Senegal y en Australia. En nuestra región, seguimos luchando, seguimos trabajando con distintos países para poder contener la plaga que se encuentra en situación de emergencia. Seguimos colaborando con especialistas de todo nuestro continente americano, pero con también investigadores de otras partes del mundo. En Sudamérica necesitamos aún más recursos y mucha más investigación para mejorar nuestro sistema de manejo preventivo. Gracias.
And here is Clara Therville to give more details about our comparative locust governance study that's supported by the Foundation for Food and Agricultural Research.
Hi, my name is Clara Therville. I'm a geographer interested in environmental governance. I'm currently working with Arianne and Marion to identify institutional barriers to sustainable locust management. While the majority of research on locusts has focused on natural science, the origin of locust outbreaks can be both determined by ecological conditions and by organizational factors. That's why we really need to pay more attention to these social variables. In our work, we compared main strengths, barriers, and challenges in terms of governance across 4 case studies: the desert locust, the South American locust, the Australian plague locust, and the Senegalese grasshopper. We use a methodology combining interviews and participative workshop organized side by side with our regional partners, SENASA and INTA in South America, the Australian Plague Locust Commission in Australia, or Acridid in Senegal. This work conducted us to assume that, along with this 2-stage ecological dynamic of locusts between recession times and crisis, governance is also characterized by the 2-stage cycle. The different case studies illustrates how, in a diversity of contexts, locust managers try to maintain coordination and trust among stakeholders over very long time frames and across a very large spatial scale. While regional workshops represent a great occasion for practitioners to develop some reflexivity regarding governance issues, our work at the global level is an opportunity to share experiences regarding organizational challenges in locust management. Thank you.
And last but not least is my PhD student, Douglas Lawton, who's going to share some of the recent modeling work that he's been doing with the desert and Australian plague locusts.
To understand what causes a locust swarm, we must understand what happens at the individual locust level. This is because the swarm is a product of interactions between individuals inducing a behavioral switch called gregarization. However, forecasting and predictive models of locust swarms rarely account for the multi-scale nature of these swarms. For my PhD, I am investigating the multi-scale ecological underpinnings of locust swarms. The key takeaway from my current research is that if we account theoretically and statistically for the multi-scale nature of locust swarms, we can dramatically improve forecasting efforts globally. For example, with the help of my collaborators in Australia at the Australian Plague Locust Commission and the University of Queensland and CIRAD in France, I am currently modeling the spatial hierarchy of preceding vegetation growth and outbreaks of 2 locust species, the desert locust in Africa and the Australian plague locust in Australia. We show that this relationship varies spatially between geographic regions and temporally between seasons. Current forecasting methods do not take this variation into account. Therefore, this endeavor increases both the theoretical and practical understanding of locust swarms. Thanks.
Thank you to everyone who contributed to this talk, to all of our tremendous GLI partners and collaborators around the world, and to everyone else for taking the time to listen to this talk. We hope we have highlighted the importance of understanding the many ways in which people and locusts are connected, as well as the value of having a global network to connect ideas, people, and organizations. If you're not already part of the GLI network, but would like to be, please email Rick Overson at roverso@asu.edu. Thank you.